Hello, IB Biology students. This is Ms. Sheely with your next installment of video lecture notes. Today we're going to look at 5.4, Cladistics. So the first thing that we need to know is that a clade is a group of organisms that have evolved from a common ancestor. A clade includes all the species that are alive today, as well as the common ancestral species and any species that evolved from that ancestor but are now extinct. And if you look at this grouping, the entire grouping, here looking at the entire grouping, this would be the common ancestor, so they would all be one clade. But if we look at the three different colors as being individual clades, the blue would be a clade because here is their common ancestor, and the pink or red would be a clade because this is their common ancestor. But the green would not be a clade because although this is their common ancestor, the green does not include the blue in the overlap of the colors. So you have two clades, small clades within this large group that have been highlighted. The evidence for which species are part of a clade can be obtained from morphology, but also from, which is how an organism looks, but also obtained from the base sequences of a gene or the corresponding amino acid sequence of a protein. Sometimes it's really hard to tell who evolved from a common ancestor, especially if you're looking just at morphology. So species that will share a common ancestor can be expected to have few differences, and species that do not share a common ancestor will have lots of differences. And this is an example of a computer printout or computer screen from a program that's looking at similarities in amino acids sequences. So all of these organisms have, you're looking at the exact same gene, and all these letters represent one amino acid. So anything that is the same is going to be colored the same, and you can easily look through and look at similarities and differences. Sequence differences accumulate gradually, so there's actually a positive correlation between the number of differences between two species and the time since they diverged from a common ancestor. That's because differences are the result of mutations, and mutations accumulate over a long period of time. We're talking about thousands to millions of years. And there is evidence that mutations occur at roughly a constant rate, so we consider these mutational differences a molecular clock. All right, looking at this cladogram here, this is been compiled using mitochondrial DNA from 3 humans and 4 related primates that have been completely sequenced. And from the differences in the base sequences, a hypothetical ancestry was formed and is constructed and shown here. And using those differences in the base sequence as a molecular clock, the approximate dates for the splits between the groups have been deduced. So the split between the European and the Japanese here was 70,000 years ago. And between the African and European Japanese branch, so this split was 140,000 years ago. And then the human chimpanzee split, so that would be here, was over 5 million years ago. So again, using those mutations in the mitochondrial DNA, we can deduce this. It's not fact, but it does. It is a visual representation that supports the evidence that is out there. All right, so traits can be analogous or homologous, and we've had some confusion with this before, and so hopefully this section will clear this up. I really like this visual. So a homologous structure is similar because of common ancestry, but they could have a different function. Our really good go-to example of this is the pentadactyl limb, where you have the 1 bone, 2 bones, many bones, and digits. So of the bats and the whales and the humans and cats and dogs and all of that. But an analogous structure is similar because of convergent evolution. And what that means is that they've evolved similar adaptations due to evolving in similar conditions. It doesn't have to be in the exact same habitat. You have rainforests in South America, and you have rainforests in Asia. So you could have a analogous structure because of different biomes around the world. But here's a really good example of looking at mammals and insects. So we have a cat, a whale, a praying mantis, and a water boatman insect. And so the legs of the cat and the praying mantis are analogous. They are used for walking on land. They've developed this adaptation in similar conditions for needing to walk on land, but they are not homologous. If you look, there are completely different structures to it. There's no evidence of common ancestry here. The same thing with the flippers. The whale flipper and the water boatman are going to use he's going to use the flipper leg, and then the whale's going to use his flipper for moving through water. So they are analogous because they have a similar function, and they've evolved because of a similar condition. But the mammals, the cat and the whale, even though they have a completely different function, they are homologous. They have a common ancestor. So even though we have a different length of the orange bone here and here, and the same for all the others, we have this very clear one bone, two bones, many bones, digits at the end. And then the insects are actually homologous structures as well for their legs due to the different pieces of those. It's very similar to the mammals and the pentadactylomes. So I hope that clears up some confusion that may have been previously with analogous and homologous. There's a lot of problems in distinguishing between homologous and analogous, as I'm sure you guys have figured out by now. And so sometimes that can actually lead to mistakes that scientists can make. And so now we very rarely use morphology and structure to develop cladograms or show evolutionary relationships. Now we really stick to those molecular clocks, the differences between amino acids and DNA-based sequences. All right, so a cladogram is just a tree diagram that shows the most probable sequence of divergence in a clade. Okay, so we're looking at one clade here, shown two different ways. A cladogram is based on similarities and differences between the species, and you have a branching point. So here would be a branching point, and here is the same branching point in the alternative view. And that branching point is called a node, and it's just a hypothetical ancestral species that split to form two or more different species. So you could have a node that actually had three species branching from it. There's evidence that the closest relatives of humans are chimpanzees and bonobos, and each species, has had their entire genome sequenced, and that sequencing has given evidence for the construction of a tree, such as the one shown here and the one shown in your book. And this tree was based on a molecular clock with a mutation rate of 10 to the negative 9th per year. And so these numbers here, we're showing the timeline of the split. So 4.5 million years ago, humans split from these primates, and 1 million years ago, the bonobos and the chimpanzees split from a common ancestor. And then this width here, this is showing you the estimated population sizes at that time. So population before the split of our ancestral species was 45,000, and then as we have split and continued to evolve, it has decreased over time. All right, so we can analyze cladograms to deduce evolutionary relationships. So the pattern of the branching is assumed to match the evolutionary origins of each species. If two clades are linked at a node, then they're closely related. If their two clades are separated by a series of nodes, then they are less closely related. Some cladograms will include numbers to actually indicate the specific number of differences. So there'll be a number at the node branch or along the branch point of the different species. And some cladograms are even drawn to scale with the length of the line indicating the size of the difference. But again, remember that cladograms are not proof. They are only constructed on evidence and made with assumptions. However, we have evidence from cladistics has shown that classification of some groups based on structure, morphology, did not correspond with the evolutionary origins of a group of species. And that's led to studying cladograms and identifying clades has led to an area of science called cladistics. And using nitrogenous base and amino acid sequences, that only really became possible towards the end of the 20th century. Some groups have now had to be reclassified. This is very time consuming and can be disruptive to biologists and other scientists working with this information. But those new classifications are likely to be closer to a truly natural classification, and everyone is willing to suffer through the time-consuming and disruptive part to get to that more like the truth information. And I have an example for you. So that basis has actually led to falsification of some theories, with one theory being superseded over another. And the example that I have for you is the classification of angiospermophytes. So angiospermophytes were first classified into families based on morphology by a French botanist named Antoine Laurent de Jussieu. And he originally published his ideas on these families in 1789. There are now more than 400 families of angiosperms. It's crazy amount of families. A particular family that he originally proposed was the figwort family. And when he originally proposed it, he only originally opposed 16 genera or genus taxa level, based again on similarities in their morphology. But 1789 was a long time ago, and as more plants were discovered that fit these morphology similarities, this family grew to include 275 genera or genus taxa level and over 5,000 species. And that put them well into the largest angiosperm family that was in existence. And recently, a taxonomic research project compared the base sequences of 3 chloroplast genes in a very large number of species in this genera. And they actually found that the species in this figwort family, they're not a true clade. They're not all coming from one common ancestor. Instead, 5 separate clades had been incorrectly combined into one family. So again, this was using genetic information about differences in the 3 chloroplast genes. All right, so that's actually it for today's video lecture notes. I hope that this information and the information from the Click and Learn has helped you to understand cladistics a little bit more. We are going to have a few activities in class where we're going to practice this as well. Thanks so much. Look forward to seeing you in class.